So cancer immunotherapy is a combination of an entire universe of science, of different subtopics of science or health sciences like biochemistry, biology, cellular and molecular biology, immunology, pharmacology, oncology, all of this together to develop a new, I would say, state-of-the-art treatment, which is actually saving a lot of lives. So my mission today is that in the next 60 minutes, you've been exposed to a lot of information that will provide you the basic knowledge of what is cancer, where we are in the time of history in terms of cancer therapy, what are the main cellular and molecular principles of immunotherapy, and what is the outcome of immunotherapy. So I will try to discuss this topic in a manner that you don't need to understand everything about immunology, but I will remind you about specific points, specific cells and process of our immunity that are critically important to the mechanism of cancer immunotherapy. So before we start here, I would like to present you to the cancer therapy timeline, where we are in history and how cancer was treated in the past. So cancer therapy actually starts 3,000 years before Christ with the Egyptians. They were actually removing tumors with surgery or trying to treat tumors with cauterization. If you can remove the tumor with surgery, great. The problem of cancer, and that's why it's called cancer, is when the tumors start spreading to different parts that you cannot remove with surgery. And then you need to adopt a different therapeutic approach that will try to reach the tumor site and kill the tumor cells. So one way of treating advanced metastatic cancers would be using radiotherapy. Back in 1900s, radiation was just discovered, like radioactivity by Maria Curie. It actually resulted in 2 Nobel Prizes. And they could actually use radiation to shrink the tumors, to kill parts of the tumors, and the tumors would reduce in size, in volume, and then surgery would be possible. But often the tumors would recover. And then afterwards in the timeline, let's go around the Second World War here, 1940s. So the scientists or the medical doctors actually, they observed one thing. They observed that the soldiers that were returning from the battlefield in the Second World War, some of these soldiers would start having myelosuppression. Literally, the reduction of the numbers of leukocytes, circulating leukocytes in the blood, lower levels of immune cells. And often these soldiers would be more vulnerable to infective diseases. So the scientists at the time, they observed that these soldiers with myelosuppression, with immune suppression, low levels of leukocytes, they were actually exposed to the mustard gas. The mustard gas was heavily used in the Second World War. It was devastating. And one of the side effects was that it would reduce the level of immune cells circulating in the body. So the doctors at the time, they had an insight. They realized that these soldiers, they didn't have good immunity because the leukocytes couldn't proliferate. They couldn't duplicate. So there was something pernicious in the mustard gas that would be detrimental for proliferating cells, for cells duplicating, like the immune cells. And they start thinking, well, we could try to use this eventually to treat cancer. Because what is cancer? Cancer is nothing but the manifestation of the duplication phenotype. The cancer cells are just growing without stopping. So maybe we can use something in this from this mustard gas that could stop the proliferation of these cells. And actually the component in the mustard gas that stops the proliferation of these cells, or the immune cells, is called the nitrogen mustard. So the nitrogen mustard has high affinity with our DNA. And imagine, if you're duplicating the cells, you're duplicating the cytoplasm, you're duplicating the cell membrane, the organelles, the proteins, and the DNA. Every cell of our body, when it needs to duplicate, will duplicate all these things. And when it comes to the DNA, if it stops in the DNA, if it cannot duplicate the DNA, all the cells of our body have a mechanism to induce cell death, because you cannot form a cell without the DNA. So if the DNA replication is blocked or impaired or even deficient, it will induce an organized cell death program, which you know, it's called apoptosis, right? So they realized that if they use the nitrogen mustard inside of the mustard gas, the nitrogen mustard, to conjugate with the DNA of the cancer cells would not be able to duplicate the DNA. They would eventually die. And this is what happens. They were discovering here the first chemotherapy, the nitrogen mustard. The problem is that it has side effects, because if you inject this systemically, and that's the reason why they are developing a new therapy, because they need to inject this in the blood so it reaches the sites or the tumor sites that cannot be removed with surgery. So you need to put this in the blood, and it will also attack normal cells. So side effects of chemotherapy is exactly what the nitrogen mustard is doing, is attacking the DNA or duplicating cells. So immune cells, as the soldiers, they will stop duplicating and they will die. So immunosuppression or myelosuppression in the bone marrow. Low levels of leukocytes. More vulnerability to infection. Another side effect, other normal cells that are proliferating in our body, like for example the cells of our gut, the mucosas. So in the gut we have the epithelial cells that are constantly renewing the epithelium. And if they die, our gut becomes sensitive, and that's why nausea and vomiting is one of the side effects of chemotherapy or this type of therapy. And then also alopecia, the hair loss, is because the cells of our scalp, they stop proliferating, they don't produce hair anymore, so they die, and then we have hair loss. So these are one of the typical side effects of this type of therapy, right? And at some point, with all these side effects together, you need to interrupt the treatment because the patient cannot bear so much trouble. So when you interrupt the treatment so the patient recovers, the tumor will also recover. And the problem is that the tumor will come with new mutations that can eventually induce resistance to the apoptotic process. So they will no longer die in front of chemotherapy. And over the decades of the timeline here, we reached like 1980s with a new type of therapy, the target therapy. So all advances in technologies enabled scientists to better understand cancer in a cellular and molecular level. Now we can visualize the cancer cells. And we can also rank the type of molecules that the cancer cells are expressing. What molecules are related with the mutations? So the cancer has a lot of mutations that are converted in proteins. What proteins are those? Where are they expressed? In the surface of the cell. Wow, if they are expressed in the surface of the cell, externally, they're like flags. And if they are like flags, we can actually target them. Let's develop now, with all this technology that we have now, new molecules, small molecules, or even antibodies that will target these flags, these products of mutations that eventually they have functions. For example, tyrosine kinases. They are constantly signaling so the cell duplicate like crazy. They are receptors located in the surface of the cell. So now we could target that and try to block these receptors so the cells duplicate less, or even inducing cell death in the cells. So scientists come to a new era here of target therapy, which is amazing. Less side effects, because these molecules, these flags are majoritarily expressed in the cancer cells, not in the normal cells or immune cells or the epithelial cells, in the cancer cells. But there's a problem here. So it's not for everyone, not every patient will be eligible for these treatments, right? And there's another problem as well. If you're using a target therapy, you will only kill the tumor cells that are actually expressing the target. And the other tumor cells eventually there that don't express the target, they'll start growing. And then you don't have another therapy to treat them because you don't know their targets. So then in 2010, it comes to prominence here in blue, and out of the blue, by the way, checkpoint inhibitors. And what the heck is that? Checkpoint inhibitors is the immunotherapy that I want to discuss today. It's an immune therapy. So all these other therapies like radiotherapy or even surgery, you go in the tumor room, you cut it out with a knife, you cut the tumor out, or you use radiation, you target the tumor, or use chemotherapy, nitrogen mustard, or whatever compound derived from the nitrogen mustard. There are several compounds that would attack the DNA or even the cytoskeleton of these tumor cells. They are designed to attack the tumor cells, side effects in normal cells. And then target therapy, designed to target the tumor cells as well. But then checkpoint inhibitors, they are immunotherapies. They will not attack, they're not designed to attack the tumor cells. They're actually specifically designed to attack the immune cells, to boost the immune cells, to reinvigorate the immune cells, because the immune cells, they have mechanisms to attack and destroy the tumors. And this was discovered in the last 50 years. Before we didn't know that. Well, we actually knew one important information that was forgotten. And this is what I want to highlight in the next slide. Immunotherapy could have started and continued already in the 1900s, but it didn't pay off. It was forgotten. This was the scientist, I would say the medical doctor, who actually started the whole immunotherapy process, who brought the conceptualization of immunotherapy. His name is William Coley, and he's considered today the father of immunotherapy. William Coley used to treat sarcoma patients. He would try to remove the tumors from the neck, sarcomas developing the neck, with surgery. And he noticed one thing: the patients with less advanced tumors, so the tumors that he could actually go there and remove with surgery, these patients actually, they would have an infection. They would often develop a type of infection, bacterial infection, usually in their legs, which is called the erysipelas. It's developed by the bacteria Streptococcus pyogenes. So he observed that the patients with a chronic infection with erysipelas, they would often have smaller tumors with lower volume that could be easily removed with surgery, as opposed to the other patients that have more advanced tumors. They usually didn't have any sort of infection. So Coley started connecting the dots here. Well, if you have infection, the tumors are smaller and easy to operate. Why not collect this infection or the product of this infection from these patients and give to the ones that don't have infection? They have more advanced tumors, so the tumors eventually would shrink and I can also treat them. So he started doing that. So Coley did exactly that. He collected some of the bacteria in one of the patients and started growing this bacteria in a petri dish. And then he extracted toxins from this culture, what we called Coley's toxin. And he literally started injecting this freaking toxin in the patients coming to treat the cancer. And the patients would have extremely high fever, like over 40 degrees and for days. Like, it was very aggressive. But guess what? The tumor starts shrinking to the degree that he could remove it. Some patients, the tumors would shrink so much that he didn't even need to remove with surgery. It would completely disappear and will never come back again. Unfortunately, other patients, they would die of fever, because what he was seeing there is exactly some of the side effects that we see until today with immunotherapy. When you induce infection or when you harness the immunity, the side effects is like a systemic inflammation. It could even kill the patient. So he started losing some patients with this approach. And at the same time, by his misfortune, radiotherapy came with 2 Nobel Prizes. And you could literally see that if you would radiate the tumors with X-rays, the tumors would literally shrink and you could remove it. But often, the tumors would come back. With this therapeutic approach, with the Coley's toxin, harnessing immunity, if you could remove the tumors will not come back. So the medical society at the time became a little bit skeptical with Coley's approach. And they said, hey, look at these patients. We don't get that, right? You're inducing fever. A lot of patients are dying. Okay, you could reduce we don't know why you're reducing the tumors, but we know with radiotherapy what's happening. So we are safer with radiotherapy. Let's continue with that. And that's why Coley was forgotten. And everything continued with radiotherapy, chemotherapy, target therapy. But at the same time, back in 1950s, a lot of discoveries start emerging in the fields of molecular medicine. And these discoveries actually resulted in Nobel Prizes that created the field of immunology. So immunology is actually a very, I would say, it's a new science. It's a young science. We only know like a few immune cells with all their functions and effector abilities, and there is still a lot more to discover. And this all came to prominence with Nobel Prize discoveries, like the antibodies, what are TCRs, what's the antigen presentation, what is an MHC. MHC, one of the most important molecules of our immunity, the major histocompatibility complex, discovered by Baruch Ben-Asseraf. And I wanted to emphasize the MHC because I will show the importance of this molecule here in the cancer immunity process, which plays an important role for cancer immunotherapy. So back in 1950s, with the studies of Baruch Ben-Asseraf and other scientists at the time that shared the Nobel Prize, it came like the genes of the immune response. So the immune system was exposed again. Now we have scientific proof of our defenses that can protect us against infection and cancer. And then Lloyd Old came as well. Lloyd Old was Baruch Ben-Asseraf's student, a PhD scientist, that he was the one who did the first translational immunotherapy approach to treat cancer. How? Good question. Lloyd Old went back to the studies of William Coley, and he said, like, there's something here. I need to look back at these studies because it was forgotten, and this is very interesting, actually. William Coley had a daughter, which is also, by the way, a medical doctor, Helen Nautz, and she actually, over the course of her life, after Coley's Toxin was completely forgotten, Helen continued the study of her father, and she got great results, and she was trying to disseminate these findings, and nobody was listening to her. But Lloyd Old, so interested in immunology, and he went to these studies and said, hey, all of this makes sense. So what he did, he tried to reproduce what William Coley was doing with the sarcoma patients. He tried to reproduce that in a lab scale using mice. He used, instead of the Coley's toxin, he used a vaccine that it's to prevent tuberculosis, the BCG vaccine, right? Developed by French scientists. The BCG vaccine was used by Lloyd Old to harness the immunity of mice, and he could actually shrink the tumors in these mice by using a tuberculosis vaccine. And he started like postulating the first steps of cancer immunotherapy, which is like we have immune cells that respond to this type of bacterial or intracellular infection, which is a similar type of immunity that fights cancer. So that was the first finding that he could reproduce in the lab using a preclinical approach, and he translated this vaccine to treat bladder cancer patients. It worked so well at the time that the BCG vaccine to treat tuberculosis, to protect against tuberculosis, is still the first line, is one of the first lines to treat bladder cancer nowadays. Absolutely successful research. So he was exposing here now in a more applicable manner how the immune system can fight cancer and bringing awareness for that. All the scientists now start looking at these works together. And he, together with Helen Nautz, he created the Cancer Research Institute in New York, from which a lot of important names learned immunology and applied immuno-oncology research to develop immunotherapy today. So a lot of Nobel Prize coming from that nursery of scientists. So during this golden age, with all these brilliant minds putting up together to look into the immune system and understand how it correlates with cancer, it comes Frank McFarlane Burnett, who is a Nobel Prize winner, by the way. He postulated the conception of the cancer immunosurveillance. Cancer immunosurveillance is nothing but the ability of our immune system to constantly check different organs and tissues and try to find carcinogenic process, so cells turning to cancer cells and eliminating them. This is happening all the time in different parts of our body, so we can very much develop cancer at any point of our lives, even now. And our immunity is so important. That's why you need to take care of your immunity, because they're so important to constantly check for carcinogenic processes happening in different tissues and immediately eliminate them, right? Just to illustrate this, if you, for example, you notice that you have a spot growing here in your skin, usually it will not be a symmetric spot. Usually it looks like an omelette. You pretty much probably saw this happening at some point in your life, and then this spot at some point disappeared. What happened actually? This spot was nothing but a group of melanocytes that start growing there. Melanocytes, they are the cells from our skin that produce melanin. And when they are all together growing in colonies, you can actually spot them. So we have these round-shaped spots. They are melanocytes, but they're not growing too fast. They're actually not even growing. So the immune system just ignored them. And that's why we have those spots. But some of them, they have irregular shape. They actually grow faster. They have some mutations. And the immune system has the ability to identify these mutations or whatever these cells are doing there. And the immunity will eliminate them. So this is the immunosurveillance process. And then Lloyd Old, with this knowledge, with this idea of the immunosurveillance, he postulated what we call the 3 E's of cancer immunoediting. Our immunity is constantly patrolling and finding carcinogenic processes and eliminating cancer cells. So here we have the normal tissues. And eventually, at any stage of our lives, could be now, could be yesterday, could be in 2 weeks, we are exposed, for example, to environmental carcinogens or to radiation or viral infections. Some viruses, they have the ability to integrate the DNA of our cells and induce mutations that will activate a proliferation program. Or chronic inflammation. So there are some certain types of inflammation that they are not good and they might suppress the local immunity, and this will favor the cancer cells to grow there. Inherited genetic mutations. So have you ever heard about the oncogenes? Yes, these are the genes that are dormant. They are bad genes. If they wake up, they might induce cancer. They activate proliferation. In our normal cells, they become cancer cells. So we don't want them to wake up. But if they have some mutations, these mutations will actually wake up these genes earlier and constantly, and they will induce proliferation of these cells. Oncogenes, they can be inherited, they could be new mutations, and they can induce cancer. So here we go. All these different causes of cancers here, they will induce the carcinogenic process and the tumor cells start growing. Remember about the spot in our hand, irregular shape. At some point start growing, in a few days it will die, right? So here the tumor cells start growing and they release this what we call danger signals because they have mutations. They start secreting things that are not normal. And our immunity starts spotting this. It's like, okay, there's something abnormal here. So what we call tumor antigens, they are expressed here. And our immune cells are recruited. They identify that, start fighting these cancer cells, eliminating them. And then you get the tissues back to normal. This is the immunosurveillance. Turns out that at some point, if the cancer cells are stimulated by these carcinogens or constant radiation or whatever life habits you might have that will induce mutation of these cells, some of these cumulative mutations might give resistance to these cells against the immune system. So all this immune pressure becomes like irrelevant because the tumors will not die. They might not grow too fast, but they will not also be eliminated. And that's what we call equilibrium stage. So at some point here, if the immunity gets stronger or lifestyle, I don't know, it will bring tumors back to the elimination. But depending also otherwise of poor lifestyle or different other factors that we are exposed to, or even aging, when the time that the immunity naturally gets weaker, these tumor cells here, they will overcome the equilibrium stage. They go to the escape stage. And the escape part of this whole story here is the most disgusting stage of the cancer. Because these cancer cells here, they have so many mutations, adaptive mutations, that will not only resist the immune pressure, but they start manipulating the immune cells to work in the tumor's favor. So some immune cells like macrophages, they will start recruiting new blood vessels there so the tumors can escape to different sites, so metastasis. Cancer cells can induce or manipulate our immunity to induce metastasis. Or the cancer cells can recruit the fibroblasts. We have fibroblasts. Fibroblasts are the ones producing collagen that makes our tissues and organs more sturdy, right? So the collagen there now start being produced by these cancer-associated fibroblasts to actually produce a shield, to shield the tumors against the lymphocytes that are coming there to attack the tumors. This is horrible. The tumors, they start creating a fibrotic wall that will shield them against the poisons of our immunity. And then the immune cells cannot kill the tumors anymore. And then they will escape using the macrophage to create new blood vessels. This is disgusting. It is extremely difficult to treat the cancer at this stage here. And the most problematic part here is the lymphocyte, because the lymphocyte is the only cells, I would say, like T lymphocytes, T CD8 cells, are going to learn this in the immunology lectures. T CD8 cells are the ones that attack the tumor, releasing poison, granzymes. They're literally perforines. They create holes in the tumor cells. Turns out that the tumors here, knowing that the lymphocytes are so dangerous for them, they accelerate the process that will make the lymphocytes tired. Exhaustion. The tumors start inducing exhaustion, and they help the exhaustion process of these lymphocytes. So they are so tired that they cannot fight the tumors anymore. This is very important. Keep that in mind. I will go back to this. So this is the conception of the 3 E's of cancer immune editing, right? Developed by Lloyd Old in support or grounded by the conception of immunosurveillance. It's extremely important to understand. And what is immunotherapy? Immunotherapy is a type of therapy that uses substances, like antibodies, for example, or small molecules, to modulate our immune system, not the cancer cells, but our immune system, to help the body to fight cancer. Actually, is to reinvigorate our immunity. There are different types of immunotherapies. Already in the past, even during the time of target therapy or chemotherapy, the scientists were trying to do that with what we call cytokines. So they were giving IL-2 or interferon gamma to try to boost the immunity. It has like a lot of side effects. Or adoptive cell transfer. So they would kind of expand the good immune cells ex vivo and give it back to the patients, so that this would kind of reconstitute a specific immune response against that metastatic cancer. Kind of work for some patients. Then CAR T cells is coming now to prominence to treat most of the hematological cancers. And then immune checkpoint therapies. The topic of the lecture today is the type of therapy that harnesses our immunity to treat most of the advanced solid cancers that cannot be removed with surgery. So this is the new milestone, the groundbreaking discovery. And to understand this type of therapy, immune checkpoint therapy, you need actually to understand how it works, the cancer immunity process. So to represent the cancer immunity process, we have this slide here. Initially, the tumors are growing, right? We are talking about solid tumors. So fragments of these tumors are constantly dying. Tumor cells, they will die, they will release fragments of tumor antigens. They are tumor antigens because they have mutations, right? So they have proteins that are not usually seen in our normal tissues. They are seen only in the tumor tissues. So these proteins that are products of the mutations, they can be detected by our immunity. So the dendritic cells, they are part of our innate immunity. They are already surrounding that particular tissue. And they start engulfing fragments of these dying cancer cells. And when they engulf these particles of dying cancer cells, they chop them to pieces, what we call peptides. Imagine a protein that's disrupted in different fragments. The smallest fragment of a protein would be the amino acid. And the group of amino acids constitute peptides. These peptides will conjugate with this groundbreaking molecule that was discovered back in the 50s, the major histocompatibility complex, or human leukocytarian antigen in humans. So the MHC is the responsible molecule to take part of this tumor antigen and bring them to the surface of the cell and present efficiently to the lymphocyte, the immune cell that actually fights cancer. Not other, okay, we have natural killer cells. They also attack cancer, but not very efficiently. The lymphocytes are the only ones that effectively attack cancer cells. And here we go. The T cells start growing here. They will come to this surrounding area with the antigen presenting cells, and their receptor that is called T cell receptor, the T cell receptor has affinity to the peptide that is coming from the tumor, what's called the tumor antigen, right? Not all lymphocytes will have affinity here. These dendritic cells had the blessing to find one clone of T lymphocyte of which the TCR has high affinity to this peptide of the tumor's peptide. And then when there is this high affinity, this marriage, this molecular marriage, then co-stimulatory molecules are expressed here. They are the additional batteries. Now, okay, we found a partner. We found a new lymphocyte that can fight this tumor. So let's now express the co-stimulatory molecules. They are mostly roughly called CD28 and B7. And these two molecules, I will show in the next slide, they start being expressed and the T cell becomes active. So initially this T cell is a naive T cell. They are dormant. They're just like quiet. But now they become active and they start growing. How does that happen in a three-dimensional perspective? If you look, for example, we're talking about melanoma, right? So let's look to melanoma formation in the skin. The tumors are growing here. And fragments of this tumor are released because the cancer cells are dying often. And these particles of tumor cells are captured by the dendritic cells, the surrounding dendritic cells that we have in our tissues normally. When they detect these fragments, they activate a metabolic program that would say, hey, I need to leave. And they will go to this, what we call lymphatic vessels. We have several lymphatic vessels in our tissues collecting parts of the tissues to get them checked in the lymphoid organs. For example, the lymph node. We have hundreds of lymph nodes in our body. They are the headquarters of our immunity to check infection and cancer cells. The dendritic cells go to the lymphatic vessels, what we call afferent lymphatic vessel, and they go to this lymphoid tissue, this lymph node, and they present the antigen to an eventual T lymphocyte there that has high affinity to this tumor antigen. The lymphocyte starts growing, dropping their circulation until find the tumor area and attacking the tumor. So this is what you need to understand. This is what happens. This is a cancer immunity in a three-dimensional perspective. So where immunotherapy fits in all of this? Now that you know the basic conceptions, let me show you the immunotherapy evolution and the timeline of immunotherapy evolution. It all started with this researcher here called James Patrick Allison. He discovered here the TCR, right? I just showed to you. The TCR is the receptor of the T cell that will match with the tumor antigen. He discovered that, 1982, very recent. Then he discovered the CD28, which is the battery, the co-stimulatory molecule that will give this boost, this activation boost to the lymphocytes so they can start growing and proliferating in the lymph node. And then afterwards, he discovered the brakes. Because you must agree that if you have an infection or fever, you have a lot of lymphocytes trying to fight an infection, you cannot have these lymphocytes activated forever. You want to turn them down after the infection. This is a natural process in our immunity. We do need to turn our lymphocytes off after the infection. So we go back to homeostasis so that our temperature reduces, so that we have less fever, so we recover. So this is the recovery process. It's a normal process to have shutdown buttons in our lymphocytes. Turns out that the lymphocytes arrive in the tumor area. They will try to kill the tumor, but because the tumors are resistant, they get tired. And the tumor starts seeing that the lymphocytes are tired, expressing these shutdown buttons. Which buttons are those? What kind of tools or engines? Allison discovered one of them, the CTLA4. Actually, other scientists start spotting that as well, but Allison brought this to prominence. He was one of the first ones to publish this finding. CTLA4 is one of the brakes of the T cells. When these T cells get tired, they express this molecule in the surface. If you press them, you turn off the lymphocyte. So what Allison did, he developed a neutralizing antibody. He started thinking, if I neutralize this button so that the tumors cannot press it, the lymphocyte will reinvigorate, will not turn off. And actually, it will have more time to attack the tumors. And that's what he did. He developed an antibody called ipilimumab that will neutralize this shutdown button. Now it turns out that the T lymphocyte will have more time to stay in the tumor microenvironment and attack the tumor. And the tumor starts shrinking, shrinking, and shrinking. From 1999 until 2010, Allison was working with this antibody to reinvigorate the T cells to attack tumors using preclinical models like advanced metastatic melanoma models, like mice would have been challenged with melanoma cells or fibrosarcoma cells and other different types of tumors, and he was astonished with the results. He would challenge the mice with the tumor, and usually when you have an anti-tumor drug that has some anti-tumor effect, it usually shrinks the tumor a little bit, but you can actually see still the tumor there. But for the first time, when Allison would check the mice, he couldn't find any tumors. He couldn't even believe the first time when he saw the experiments, and he wanted to repeat it. And of course, he would repeat it. And again, no tumors whatsoever with the protocol that he was following. So it was emerging, the first immune checkpoint therapy, anti-CTLA-4, removing the brakes of our T cells so they can invigorate and attack the tumors without restrictions. So the first patient that was treated with this ipilimumab was Sharon. She was like in her 30s, and she had advanced metastatic melanoma with metastases in the brains, in the lungs, and different parts of the body. She already tried all therapies. No therapy would work. And there was just this therapy coming from the lab now. And they talked with her, and they asked her, hey, would you like to try these antibodies? Like, it showed amazing results in mice, but we still don't know any effect in humans. But it's something very promising. Nothing else will work with you. And they were expecting not a long time of survival for her, like giving just a few months of life. And of course, please give me the sheets. Where do I sign it? I want to receive this therapy. If you can guarantee, or if you think that it can help me, I'm more than happy to take it. So remember, she had metastases in the brains, lungs, bones, intestines, everywhere. She received this immune checkpoint therapy, ipilimumab. She's still alive. The metastases in the brains, lungs, all disappeared. She got some side effects, of course. These treatments are not absent of side effects. Kind of like it activates autoimmunity side effects, but she's still alive. And he treated the tumors, and the tumors didn't recover. They also treat other patients with this drug. It's not a magic bullet, of course. Other patients, unfortunately, would develop more of the side effects of autoimmunity. That's what the antibody does. It removes the brakes of our immunity, and some patients might eventually develop autoimmune reactions with normal tissues. So they need to interrupt these treatments. But regardless, a lot of patients respond, and for the first time, it's kind of working the core of our immunity that would reproduce what William Coley did in the beginning. It would reactivate our immunity so that it will not only eliminate the tumor, but we will give you memory and adaptability so the tumors will not come back, because these T lymphocytes, afterwards, they become memory cells, and that will help us to prevent the tumors to come back. This is amazing. So for the first time in 2010, we are able to remove the brakes of our immunity and improve our immunity in a more specific manner to fight cancer. So we're not targeting the cancer cells. We are targeting the brakes of our immunity. And then at the same time, this was a golden decade as well. Another researcher from Japan, Tasuku Honjo, he discovered another brake of our immunity, which is called PD-1. It's even more important than CTLA-4. It's unbelievable. And they developed another antibody that would block this brake. So in combination, the anti-PD-1 and anti-CTLA-4, these antibodies, when they block these shutdown buttons both together, it removes the brakes of our immunity so that the patients respond even more. So now we have like literally, or roughly, I would say 52% of patients long-term survival, and up to 30% of patients can even achieve complete response, complete remission of the tumors, at least in melanoma case, which is a responsive type of tumors. There are types of tumors that do not respond that well for this type of therapy, but these are the baby steps that we are taking now towards of using the immune system to educate our lymphocytes to properly fight the tumors. It's unbelievable. So that for given to those results, this new groundbreaking milestone in cancer therapy timeline, that these two researchers received the Nobel Prize in 2018. So now let's recap the entire cancer immunity, but put together here the conception of cancer immunotherapy. So now you understand exactly what's happening here. Again, the tumors are releasing the dead fragments. Remember from the previous schematics. And then these fragments are captured by antigen-presenting cells that will chop them to pieces into peptides to present them into the MHC molecule, Nobel Prize topic. This MHC molecule will present the tumor fragment to the lymphocyte through the engagement of the T cell receptor here. Look, it's so beautiful. Great discovery here from James Allison. And this marriage will engage the T cell to become active. So we have like here the co-stimulatory molecules that start being expressed and creating these immunological synapses here that will activate the T cell. The cells start proliferating, creating an army to attack the tumors. The problem is that after some point, these T cells get tired. It's a normal process of our immune cells. They need to get tired, so we reach the homeostasis and the fever will go down. It's after a certain period of time that they remain active, they will get tired and they will express the shutdown buttons. In this case here is this red molecule, it's called CTLA-4. Turns out that the tumors might influence the dendritic cells or even themselves, and they will press the CTLA-4 here to terminate the activation and proliferation of our T cells. As a result, the tumors will recover. And that's how they escape our immune suppression. And the third E of cancer immune editing. The CTLA-4 is an immune checkpoint because it's the brake. So it's a checkpoint of our immunity. It turns off. But then you bring this inhibitor. So immune checkpoint inhibitor will inhibit the checkpoint, will inhibit the brake. It blocks the brake. And the T cells will now persist in their activation and proliferation to terminate the tumor or to induce tumor destruction. That's what happens. So we recover our immunosurveillance by removing the brakes of our immunity. It's very beautiful. This is really Nobel Prize, right? So how can we say that immune checkpoint therapy is beneficial to the patients? So let's understand here. How can you actually see a great result of a cancer treatment, in this case, immune checkpoint therapy? Usually, the clinicians, they look at these results by interpreting a specific type of curve that we call survival curve. You might be familiar with the survival curve or not, but it's very easy to understand. Usually, the curve represents the time that the patient remains alive, or the group of patients remains alive. This is represented in percentages, right? So in the left axis here, the y-axis, we have the survival frequency. Every time that the curve has a drop, it's because there's a death event. So the faster the curve drops, the lower the survival, because we have more death events in a shorter period of time. But the longer the time so that the curve drops, the longer the survival. So there's something there impairing the patients to die. So this is how we interpret these survival plots. Usually the ones that are dropping faster, what we call high risk score, and the ones that take longer to drop, the curves that take longer, are the survival curves with a long period of follow-up. They are low risk score. So when it comes to these immune checkpoint inhibitors, the ones that will block, for example, PD-1, the name of this molecule, this drug, is called nivolumab. It's an anti-PD-1. It will target and inhibit the PD-1 brake, right? So nivolumab will target this brake. in comparison with docetaxel, it's actually a chemotherapy. It's a chemotherapy that will attack the cytoskeleton of cells that are proliferating. It has side effects, right? So you can clearly see here that the patients that are receiving this nivolumab, it takes a little bit longer for them to drop in the survival curve as opposed to docetaxel. So if you look into the 12-month survival rate, you still have 42% of patients alive, as opposed to docetaxel, you only have 24%. So here we can roughly say that there is almost 100% of survival improvement in the 12 months. From 24, you go to 42. It's almost 100% of improvement. And then when it goes to 24 months, you still have roughly 30% of patients still alive with nivolumab. In these cases, advanced metastatic melanoma patients. But then another study shows the combination of nivolumab with ipilimumab in a 36 months of follow-up, at least 58% of patients are still alive. This was never seen before. Really. Usually with chemotherapy, most of the patients already died, like with 36 months follow-up. And here, with the combination of nivolumab with ipilimumab, which is anti-CTLA-4, you still have 58% of patients alive. And these studies are still going on. We still have the follow-up of these patients, now with 5 years, 6 years, 7 years, and this curve is now achieving a plateau, what we call, they are not dropping anymore, so the patients are actually responding. They achieved the complete response of most of them. So this is unbelievable. It was never seen before. And we still have like some critical unsolved questions regarding these treatments because they are new, right? So Nobel Prize for that came in 2018. And now we are trying to work, we scientists are trying to work in these two main questions that are not solved yet. Why some cancer types still don't respond to immune checkpoint therapies. For example, metastatic ocular melanomas, they are completely refractory to these treatments. These patients do not respond to immune checkpoint therapies for some reason. And why some other patients that initially respond, they will relapse the treatment. So these are the cases here of inherited resistance to immune checkpoint therapies or resistance that is acquired during the treatment. And these are the main puzzles that we scientists are trying to work on. We need to understand what kind of resistance is involved in these patients so we can develop new treatments that will block the resistance, and then we come with immune checkpoint therapy to release the brakes and the patients will respond. This is what we are trying to do nowadays. This is where the next decade of immune checkpoint therapy will go, in the development of adjuvant combinatory treatments to unleash the full power of immune checkpoint therapies by blocking resistance. Beautiful, isn't it? Right, it's not difficult. You can really start reading about this now. You understand what is a dendritic cell, what is an MHC molecule. You understand what's the role of presenting the antigen to the T cell receptor of the T lymphocyte. Which lymphocyte? TCD8 cells, right? The ones that mostly attack the tumors. But we also need the TCD4. Read about the TCD4. They are the ones orchestrating the entire cytokine milieu in the tumor microenvironment. Okay? This is a very beautiful subject of immunology. And I also have a research group in cancer immunotherapy. So we try to develop tools that will predict if the patient responds to this treatment. Yes, this is called prognostication. And also we develop adjuvant therapy. So we are also trying to develop a discovery mechanism of resistance to block them so the immune checkpoint therapies respond better. So thank you very much for your time watching this video. If you have any questions, don't hesitate to ask in the comments below. I'm more than happy to come here, answer for you, or even make a brief video to explain specific topics. I know it's pretty much impossible to cover the whole thing in just one video, but I'm more than happy to come here and expand the discussions with you. And if you really like this channel, you would like me to continue doing videos like this, you can really help. There are different ways to help. For example, you can share the channel, you can subscribe, you can comment on the videos, making the channel more active, you can like the videos. All of these activities help the channel to grow. And I really believe that everyone has the right to know how our immunodefenses work. And this is sort of a mission that I have in my heart. And it would be great if people would not only know how our defenses work, but what are the top science coming out of the research labs now that would provide with the best treatment to treat advanced diseases like cancer, for example. Everyone has the right to know that. It's my mission to bring this awareness. So join with me in this mission. I'll see you in the next video. Bye bye.